Western blotting Part 1 Protein Isolation. In this animation, we will introduce you to an experimental technique called Western blotting. Western blotting is a technique that allows researchers to detect changes in particular protein of interest, such as changes in concentration, addition or subtraction of small chemical modification, and changes in interactions with other proteins. In the Western blotting procedure, proteins are first isolated from the rest of the cell components to extract proteins from cells. Researchers use chemical-based and/or physical-based method to disrupt the cell plasma membrane and release proteins inside it. Chemical-based lysis use various types of detergents to make protein accessible. For example, weak detergents are used to extract water-soluble proteins inside the cell without disrupting intracellular membrane compartments. Strong detergents are able to solubilize or make available the proteins found within all membranes. Following lysis, cells and their contents are spun in microcentrifuge. This results into an aqueous supernatant which contains solubilized proteins, and a pellet which contains membranes, organelles, nucleic acids, and any remaining insolubilized proteins. The supernatant, now called a protein lysate, is separated from the pellet to use for Western blotting. The common type of protein lysate that researchers prepare is called a whole cell lysate because it contains all of the proteins within a cell. Two other commonly prepared types of lysates are nuclear and cytoplasmic lysates, which contain only nuclear and cytoplasmic proteins respectively, often prepared to study the regulation of protein localization to these two compartments.
This section explains the basics of Western blotting, a powerful method for detecting individual proteins in complicated mixtures. The method starts with protein extraction and measurement. A lysis buffer breaks cells to release protein from tissue and cells. Once extracted, protein must be quantified to guarantee uniform loading across samples. Protein concentration is measured correctly using Bradford or BCA tests. SDS page separates protein by size after extraction. Butagen SDS denatures protein and covers them with a uniform negative charge. Unfolding them, the protein samples are put onto a polyceramide gel and electric current is administered. Sign-based separation occurs because smaller proteins travel faster through the gel matrix than bigger proteins. After separation, proteins are transported from the gel to nitrocellulose or PVDF membrane. Electric current transfers protein onto the membrane, making them easier to detect. To avoid non-specific binding, the membrane must be blocked before antibody probing. The membrane is blocked by soaking it in BSA or non-fat milk. This process plugs membrane gaps to ensure antibodies bind solely to target protein.
The protein of interest can be detected through direct or indirect approach using antibodies. These antibodies have been validated for use in Western blotting and are either species-specific, class-specific, or isotype-specific in order to prevent cross-reactivity. For direct detection, the target protein is tagged by a single antibody that is conjugated to a reporter molecule, such as biotin or fluorophore. With indirect detection, the membrane will be incubated with unlabeled primary antibodies that will bind specifically to the target proteins. After washing, the primary antibodies will be captured by secondary antibodies. The secondary antibodies have typically been raised against the immunoglobulin class or subclass of the primary antibodies. The secondary antibodies are also labeled with enzymes such as horseradish peroxidase that produce a signal when treated with substrate. Colorimetric substrates produce a colored product visible on the membrane, while chemiluminescent substrates produce a light that can be detected by X-ray film or a CCD camera. Fluorophores, however, only require an appropriate imaging system to provide highly sensitive results. The intensity and volume of the band is directly proportional to the concentration of the target protein. The now visible band of target protein can then be compared to the corresponding band of molecular weight markers for further confirmation.
Now, let's talk about the application of Western blotting. In research applications, Western blotting plays a vital role in various research fields, particularly in understanding diseases and molecular mechanisms. One significant application is in disease diagnosis. For example, Western blotting is widely used to confirm HIV infection. The test detects specific viral protein, providing a reliable diagnostic result. Another key area is cancer research, where Western blotting helps analyze protein expression patterns associated with cancer progression. Other than that, in biotechnology and pharmaceutical industry, Western blotting is essential for quality control. It ensures the correct expressions and purity of therapeutic proteins, such as insulin or monoclonal antibodies. This step is crucial for maintaining safety and efficiency in biopharmaceutical. In conclusion, Western blotting is a powerful and reliable technique that has revolutionized molecular biology. Its ability to detect specific protein with high sensitivity and specificity make it indispensable in research, diagnosis, and drugs development, from diagnosing diseases like HIV to advancing cancer research, and ensuring the quality of therapeutic protein. Western blotting remains a cornerstone technique. As molecular biology continues to evolve, Western blotting will undoubtedly remain at the forefront, driving breakthrough in science and medicines. That's all from us. Thank you.